coming to the next part of the chapter, okay, let us continue with what? The salient features of genetic code. Okay, coming to the salient features of genetic code. We have finished 5 salient features of the genetic code in our previous part, and coming to the next one, that is 6th salient feature is genetic code is non-overlapping. Okay, it means the genetic code reads linearly. Okay, it reads linearly. There will be no any of the punctuations, as I have told earlier. Okay, it will be reading linearly. Right. Next one is the direction of reading the genetic code. The genetic code will be always read in the direction of 5 dash to 3 dash or 5 prime to 3 prime direction. Okay. If you are given with a particular genetic code, okay, you should read always the same in the direction of what? 5 dash to 3 dash. Clear? Next one. Coming to the next part of the chapter or next point, that is anticodon. Each codon has got what? A complementary anticodon. Do you remember? Each codon has got a complementary anticodon. Okay, and where exactly the anticodon for a particular codon will be present? It will be present on the tRNA. The anticodon loop of tRNA contains anticodon for each and every codon in the mRNA. Okay, the codons will be present in the mRNA, and anticodon for the particular codon will be present on the tRNA. Clear? Next point, nonsense codons. Okay, what are nonsense codons? They are also called as stop codons, as I have explained you earlier only. Okay, stop codons are there, UAA. Okay, so this is not GUA, sorry, it is UAG. Okay, then UGA. Okay, the next one is UGA. Okay, UG and A. So these are the what? 3 stop codons. Is that clear? UAA, UAG and UGA are the 3 stop codons that will be present. Right? Next one. Coming to the next part of the chapter, that is mutations and genetic code. Okay? Coming to the next part, that is mutations and genetic code. So how exactly the mutations are related with the genetic code? Okay. See, the relationship between DNA and genes are best understood by means of what? The mutation. So coming to the first question, what exactly is meant by mutation? So what do you mean by mutation? Do remember that mutation is a process in which sudden inheritable changes will be seen in the particular part of the DNA or particular DNA molecule. Okay, what is mutation? Sudden inheritable changes. Okay, not just a change, but it should be inheritable. Okay, sudden inheritable changes found in the DNA are called as what? Mutations. Okay, and how exactly the mutations occurs and what are the types of mutations? Okay, let us see that. In the mutation, first of all, okay, you can see point mutation and frame shift mutation. So what do you mean by point mutation? See, in the DNA, you can find several number of what? Okay, the nitrogenous bases. Okay, at the same time, in the RNA also you can find several number of what? Nitrogenous bases that will be present in a triplet form to make a codon. Is that clear? Like this, the nitrogenous bases will be found on the mRNA. Is that clear? But in the first codon, as you already know, always the first codon should be AUG because it is initiator codon. If AUG, okay, is altered by any one particular nitrogenous base, if AUG becomes okay GUG, then what will happen? The glutamic acid, okay, sorry, the methionine should be produced by AUG, but if it is mutated, then what will happen? It will be becoming GUG. If AUG becomes GUG, then what will happen? Instead of having methionine, okay, instead of having methionine, valine will be produced. Okay, instead of having methionine, valine will be produced. So like that, what will happen? Mutation at one particular point, okay, or at one particular at one particular nitrogenous base will be called as what? Point mutation. Okay, what is point mutation? Mutation that occur in only one codon, that too particularly in only one nitrogenous base. Okay, if the mutation occurs due to replacement of nitrogenous base within the gene, then it is called point mutation. Okay, it affects, see here, it only affects the change of that particular amino acid, but it will not affect the complete part of the mRNA, but it will affect only the part, only that particular gene, okay, only that particular codon it will affect. And other one is there called as frame shift mutation. What is frame shift mutation? It is the type of mutation that occurs due to what? Insertion or deletion of what? One or more nitrogenous bases. Okay, by means of insertion or deletion of one or more nitrogenous bases, what will happen? Frame shift mutation will occur. Say for example, if there is a word, okay, if there is a word like big, okay, in this particular word, if any one word, any one letter is added in between what? I and G, okay, that will be, okay, what will happen? B, I, E, okay, I'm going to add here E, and the last one is as it is G. If any one letter is added here, then what will happen? Frame shift will be taking place. That means here in the position of G, okay, E has been inserted, so the G will shift to the next position. Is that clear? G will shift to the next position. So the G is shifted to its next position, that is to its right-handed direction. To the next part of the position, the G will be shifted. So that is called as frame shift. Okay, and by means of deletion of I, the G will shift back. Is that clear? By means of what? Deletion of a particular nitrogenous base. If you consider I as a nitrogenous base, if you delete that, then the G can be shifted back to the left side. So this shifting of the what? Nitrogenous bases will occur by means of insertion of another nitrogenous base or deletion of one nitrogenous base. Okay, by means of deletion or insertion of one or more nitrogenous bases will lead to what? The frame shift. Is that clear? Will lead to shifting of the frame. So that is called as what? Frame shift mutation. What is frame shift mutation? I repeat here, it is the type of mutation which is caused by means of insertion of a particular nitrogenous base or more than one nitrogenous base, or by deletion of one or more nitrogenous base. So by means of insertion or deletion or one or more nitrogenous base, what will happen? Shifting of the next or previous nitrogenous bases will take place towards their right side or towards their left side. So that shifting of the nitrogenous bases or shifting which takes place is known as frame shift mutation. Okay, it is known as what? Frame shift mutation. Here, by means of that frame shift, what will happen? The change in the whole sequence of amino acid will take place. Okay, change in the whole sequence of the amino acids will take place. Say for example, I have taken only one triplet here. If you write more than one triplet continuously, and if you delete one, all the further triplets which are there, they will shift back. Is that clear? If you delete, and if you add one more, they will be shifted to the next position. That is why by means of deletion or insertion, what will happen? The frame shift will take place. Okay, by means of frame shift, the change in whole sequence of the amino acids can be seen. Okay, next one. The types of mutation. See here, in detail, we are going to study about the types of mutation in this particular slide. See, in the mutations, there are three types. Okay, in the mutations, there are what? How many types? Three types are there. Okay, which are they? First one is base substitutions. Second one is deletions. Third one is insertions. Base substitutions, then deletions, then insertions. These are the three different types of mutations are present. Okay. Further different types of mutations are also present, but for your level, okay, for the level of NEET and the CET, up to these three different types of mutations are important. Okay. In that, coming to the first one, base substitutions. What exactly is meant by base substitution? See here, single base substitutions are called as point mutations, okay, as we have discussed earlier, right? In the point mutation, what will happen? A is replaced by G there, or else one particular nitrogenous base will be replaced by another nitrogenous base, okay? That is why it is called as point mutation or base substitutions. Point mutations are also called as base substitutions. Is that clear? Then, you might have already heard about this particular example. In case of sickle cell anemia, the disease sickle cell anemia, what will happen? Glutamic acid will be replaced by valine. Okay? Due to replacement of glutamic acid by valine, okay, what will happen? It will lead to a disease called as sickle cell anemia. Okay? In the sixth position of beta globulin chain of the hemoglobin, okay, what will happen? Due to replacement of one particular nitrogenous base, okay, what will happen? The mutation will be resulted. Due to that particular mutation, okay, the GUG will be turned into GAG. So that will ultimately lead to what? Okay, GAG will turn into GUG, so that glutamic acid will be replaced by valine. You have already studied this particular example in the previous chapter, okay, that is principles of inheritance and variations. In that particular chapter, we have studied about the sickle cell anemia. We have discussed even about the sickle cell anemia, right? Next, see one important point. Point mutations are most common type of mutations, and there are two types in the point mutations. Okay, point mutations are of two types, or else base substitution mutations are of two types. The first one is transition, second one is transversion. Okay, very important points to be noted here. In case of base substitutions, there are two different types in the base substitution mutations. The first one is transition and second one is what? Transversion. So coming to the first one, that is transition. What exactly is meant by transition? What exactly happens in the transition type of mutation? See, it is the type of mutation in which, okay, a purine is very important. See here, a purine is substituted with another purine, or when a pyrimidine is substituted with another pyrimidine. Okay, if purine, that means if A is substituted by, okay, A is a purine, adenosine is a purine. If the A is substituted by G, okay, that means both are purines here. If A is substituted by G or else G substituted by A, then that type of mutation is called as transition type of mutation. Okay, it is called as what? Transition mutation. Is that clear? What exactly happens in the base substitution? If one base, if it is a purine, and if the purine is replaced by another purine, if one purine is replaced by another purine, or a pyrimidine is replaced by another pyrimidine, then it is called as transition. Very important point here. If a purine is replaced by purine and pyrimidine is replaced by pyrimidine. If A is replaced by G or G is replaced by A, or else T is replaced by, okay, T or U, it is replaced by, okay, the C, then it is called as what? The transition type of mutation. Okay, it is called as transition type of mutation. Next one, transversion. Okay, it is the another type of base substitution or point mutation in which, okay, purine is substituted by pyrimidine or pyrimidine is substituted by purine. So, if purine is replaced by pyrimidine or pyrimidine replaced by purine, then it is called as transversion. In the first one, transition, what will happen? The substitution occurs between purine and a purine and pyrimidine and a pyrimidine. But in case of second one, the substitution occurs between purine and pyrimidine. Is that clear? So that is the major difference due to this particular change. Okay, the name is given as transversion. Okay, transversion will take place. Okay, what is transversion? It's a type of mutation in which, okay, a purine is replaced by pyrimidine. Okay, purine is replaced by pyrimidine, right? Or pyrimidine is replaced by purine. So this is what the type of mutation. Okay, first type. Next one, deletions and insertions. Okay, what is deletion? Okay, see here, a deletion resulting in frame shift mutation that results when one or more base pairs are lost from the DNA. Okay, when one or more base pairs are lost from the DNA. See here, see in the figure above, I'll draw the figure. See, if this is mRNA or this is the DNA, and here what are there? Nitrogenous bases A and U, then G, then C, then A, like this, somewhat it is there. If the G is deleted here, then what will happen? C and A will shift back. Okay, so that itself is called as what? Deletion mutation. By means of deletion of single base or else multiple bases, okay, what will happen? The frame will be shifted to the left side. Very important point. It may be asked in your NEET examination or CET. By means of deletion, the frame will shift towards which particular direction? Okay, it will be shifted to the left direction or left side, right? And if one or two bases are deleted, okay, translation frame is altered and resulting in the garbled message and non-functional product. Okay, by means of deletion, what will happen? Frame will be shifted to the left side, and when the frame is shifted to the left side, then what will happen? The complete sequence of the amino acid, okay, the complete sequence of the amino acid will be changed. When complete sequence of the amino acid changes, then what will happen? It will ultimately leads to the formation of a completely different what? Protein, okay, or polypeptide chain. It will lead to formation of completely different polypeptide chain. Is that clear? When it leads to the formation of completely different polypeptide chain, then it may give rise to proper protein, or it may ultimately lead to what? Formation of what? A grambled or else gravelled product, okay, or else a particular inactive, okay, inactive protein or inactive, or else the waste product will be produced. Clear? So that, see, a deletion or, okay, one or two bases will lead to what? The translation frame that is altered and resulting in what? A grambled message and non-functional product. The product whichever is formed by means of deletion of one or two bases will lead to production of what? The non-functional product. Next one, if three bases are deleted or more bases are deleted, leaving the reading frame shift intact, then what will happen? Deletion of one or more codons result in protein missing in one or more amino acid. Okay, it may be deleterious. Okay, next, coming to the next one, that is insertion mutation. Here, what will happen? By means of insertion, or else in this type of mutation, okay, the mutation occur due to what? Addition of base pairs. Okay, by means of addition, what will happen? The frame will be shifted to the right side. Okay, the frame will be shifted to what? Right side. Is that clear? And ultimately, combination of insertion and deletions lead to a variety of outcomes. Okay, and in case of insertion, when the frame is shifted to the next one, then also what will happen? The complete sequence of amino acids can get changed. Okay, complete sequence of amino acid will get changed. Okay, similarly, okay, very similar to that of what deletions, the insertion will also be functioning. Okay, very similar, but in case of deletion, okay, the frame will be shifted to the left side. In case of insertion, the frame will be shifted to the right side. Clear? Coming to next and important part of this particular chapter, that is the tRNA, which is called as adapter molecule. Here you can see the structure of tRNA. So tRNA adapter molecule. What is tRNA? See, the tRNA is called as sRNA or else soluble RNA. Very, very important point to be noted here. The tRNA can also be called as sRNA or else soluble RNA because it is soluble in nature. Is that clear? Next one, it acts as adapter molecule. Again, an important point to be noted here. Since it acts as adapter molecule, the tRNA can also be commonly known as what? Okay, adapter molecule. Since it is known for what? Its adapter nature, it can be called as adapter molecule. Next one, see, tRNA has an anticodon loop, okay, that base complementary to the codon. Okay, anticodon loop. See, in this diagram I am showing here, this is the loop which is called as anticodon loop. And in the anticodon loop, what will be there? Anticodon will be present for a particular codon. Okay, as we have already discussed in the previous part. Okay, in case of the salient features of genetic code, we have discussed. Okay, tRNAs will have, okay, the tRNA molecules will have anticodons. Okay, and where exactly the anticodons will be present on the tRNA? In the what? Anticodon loop of the tRNA. And which particular anticodon will be present? The anticodon of a particular codon will be present on the tRNA. Right? Next one. It has an amino acid acceptor end, okay, which binds with the amino acid. See here. Amino acid acceptor end, that is 3' end of the tRNA. Very, very important point to be noted here. Okay, very important point to be noted, that is the 3' end of the tRNA. Okay, the 3' end of the tRNA will have what? A particular site where amino acid is attached. Okay, that is called as amino acid acceptor end, or it is called as amino acid binding site. Okay, the 3' end of the tRNA will have amino acid binding site. For that sake, the amino acid will be attached to this particular end of the tRNA. Okay, next one. Each tRNA bind with specific amino acid. Very, very important point to be noted. Each tRNA, that means a particular tRNA will bind to a particular amino acid only. Okay, it depends upon what? The anticodon which is present in the anticodon loop of the tRNA. Is that clear? Each tRNA will specifically bind with specific amino acid, which depends upon the anticodon that is present in the anticodon loop of tRNA. Is that clear? And by means of this nature, there are 61 different types of tRNAs are found. Because 61 what? sense codons are there. Okay, 61 sense codons are there. Due to 61 sense codons, 61 different types of what will be present? tRNAs will be present. Next one. One specific tRNA with anticodon, okay, UAC is called as, one specific tRNA with anticodon UAC, okay, it is not UAC, sorry for the mistake here, it is, okay, UCA. Okay, the point is correct here, students. Sorry for the disturbance. One specific tRNA with anticodon UAC. Okay, anticodon UAC. Actually, the codon for this anticodon will be A, U, and G. The codon will be AUG. And for this codon AUG, the anticodon on the tRNA will be what? UAC. And this particular tRNA which has got anticodon UAC will be acting as initiator tRNA. Very important point. The initiator tRNA will have what? UAC as its anticodon, because the initiator codon is what? AUG. Okay, initiator codon is AUG. That is why the initiator tRNA should have the anticodon, that is U, A, and C. Next one. There is no tRNA for stop codons. Very important point here. Okay. There are 3 stop codons, as you already know, that is UAA, UGA, UAG. Okay. These are the 3 stop codons. And why these particular codons are called as stop codons? Because they do not code for any of the amino acids. And if you ask the question, why they do not code for any of the amino acid, the answer is because they don't have any of the tRNA, okay, which has got anticodon for these and which can carry the amino acid for these particular codons, right? That is why there is no tRNA for stop codons, okay? And next point, the secondary structure of the tRNA will be clover leaf like. Okay, the secondary structure of the tRNA will be clover leaf like. That is why you can call this particular tRNA structure as clover leaf model. Okay, clover leaf model of tRNA. Okay, and here clover leaf model of tRNA, I'll just draw it simply here. Okay, it will be having the structure like this. Okay, this is what the clover leaf model of what? tRNA. Okay, and the actual structure of tRNA is compact and looks like L shape. Okay, here the actual structure will be there and it will be looking as L shaped. Okay, it is a diagrammatic, and if you observe the chemical structure, it will be L shaped. Okay, coming to the next part of the chapter and most important, that is translation. Okay, what do you mean by translation? Okay, what do you mean by translation? Translation refers to polymerization of amino acids from what? The mRNA to form a polypeptide chain. Okay, the process of formation of, okay, you can also define the translation in other ways. Okay, that is the process of biosynthesis of, okay, the process of biosynthesis of amino acid chain. Okay, the process of biosynthesis of amino acid chain with the help of adapter tRNA and the ribosomes is called as what? Translation. Okay, the process of biosynthesis of protein is called as translation, or even you can define it as the process of formation of amino acid chains by means of decoding the coded information from mRNA. Okay, decoding the coded information from mRNA. So that can also be called as what? Translation. Okay, So the translation can be defined in varieties of ways. But ultimately, what is the meaning of translation? It is nothing but the process of formation of a particular polypeptide chain. Okay, the process of formation of a particular polypeptide chain, that is called as translation. Next, for the purpose of translation, okay, how exactly it will be taking place? About that we shall discuss in this particular slide. Right. See here, number of the number and sequence of amino acids are defined by the sequences of bases present in the mRNA. Okay. For the purpose of formation of polypeptide chain or amino acid chain, okay, the amino acid sequence should be noted. Okay, and these amino acid sequence, okay, will be present, okay, or the code for this amino acid sequence will be present in the mRNA. Okay, in the form of coded information, it will be present in the mRNA. Right, and these amino acids are joined by means of peptide bond. Okay, see here, the amino acids are joined by means of what? Peptide bond like this to form amino acid chain, or it is also called as what? The polypeptide chain. Next one, how exactly this process take place, okay, and where it takes place. Do remember, the process of translation takes place inside the cytoplasm of the cell. Okay, the process of translation takes place inside the cytoplasm of the cell. Why? Because the mRNA which is formed after translation, after transcription, it will come out of the nucleus with the help of nuclear pores. By means of nuclear pores, the mRNA will come out and it will be suspended in the cytoplasm. And the process of translation will take place in the cytoplasm of the cell. Right. And first of all, before the initiation of the process of translation, what will happen? The amino acids are activated in the presence of ATP. Okay, and they get linked to the specific tRNA. Okay, see here, what happens? Before the beginning of translation, or at the time of beginning of the translation, what will happen? Amino acylation of tRNA or charging of tRNA will take place. Amino acylation or charging of the tRNA will take place. What do you mean by amino acylation or charging of the tRNA? Very simple. Before initiation of translation, what will happen? The amino acids will get activated in presence of the ATP, and what the activated amino acids will do? They will go and bind to the specific tRNA molecules. Okay, they will go and bind to specific tRNA molecules. And that particular process of binding of activated amino acids to the particular tRNA molecules is called as amino acylation of tRNA. It is called as what? Amino acylation of tRNA. And next point, ribosome is cellular factory of protein synthesis, okay, which consists of two types. Okay, that consists of what? Two types. That is 70S ribosome is there and 80S ribosome is there, and both of these, okay, will be helping for what? The protein synthesis in prokaryotes and eukaryotes separately. Clear? And to remember, the ribosomes, okay, the ribosomes will be, okay, the ribosomes will be consisting of, okay, in their activated form, they will be consisting of a larger subunit and a smaller subunit. Okay, a larger subunit will be there and smaller subunit will be there. When both larger and smaller subunits are united, they will form active ribosome, which will be helping out for what? The protein synthesis. Okay, and here, see, we have mentioned that the ribosomes are of 70S. Okay, this 70S ribosome will be formed by means of union of larger subunit, which is 50S, and a smaller subunit, that is 30S. Here you should not add 50 plus 30, okay, because they are not the particular elements here to add, okay, they are the particular weightage of this particular what? The RNA, sorry, not RNA, the ribosomes, okay. And here 50s ribosome and 30s ribosome, when they unite together, they will form 70s type of ribosomes. Clear? And what exactly is meant by S here? The S is nothing but Svedberg's unit. Okay? S means Svedberg's unit. Okay? See, I'll repeat this particular slide again. Translation. What is translation? The process of biosynthesis of protein from mRNA template with the help of, okay, the process of biosynthesis of protein from mRNA template with the help of what? The ribosome and tRNA. Okay, with the help of ribosome and tRNA. So that is called as translation. Or simply you can say that translation is a process of formation of what? Protein from the mRNA. Okay, process of formation of protein from mRNA is called as translation. Before the beginning of translation, what will happen? Amino acylation of tRNAs will take place. Okay, amino acylation of tRNA, that you know. What is amino acylation of tRNA? Or it is also called as charging of tRNA. What exactly happens in this step? Very simple. The amino acids will get activated, and these activated amino acids will get attached to the what? Amino acid binding site of tRNA. Okay, they will get attached to amino acid binding site of the tRNA. Is that clear? That process is known as charging of the tRNA or amino acylation. Right. And do remember, the sequence of amino acids in that particular polypeptide chain or amino acid chain will be determined by the particular codons that are present in the mRNA. See here. Okay, based upon the codons that are present in the mRNA, what will be formed? The amino acid chain will be formed. And coming to the next one, the process of translation. Okay, how exactly it will be taking place, right? The process of translation takes place in 3 steps again, as in case of transcription, which has got 3 steps. Okay, similarly, the translation is also having 3 steps. And the first step of translation is initiation. Okay, the first step of translation is what? Initiation. What exactly is meant by initiation? Okay, see, it is the first step of translation in which, okay, the smaller subunit of, okay, the smaller subunit of ribosome, okay, will get attached to what? The mRNA. It is the first step of translation in which, okay, the smaller subunit of, see here, the smaller subunit of ribosome and mRNA both will get attached to one another. By means of attachment of smaller subunit of ribosome and the mRNA, initiation will be taking place. Okay, and the smaller subunit of ribosome will attach to the mRNA in a specific region where the codon AUG is present. Okay, the ribosome, very important point to note here, the smaller subunit of ribosome will bind to mRNA in the first step, no doubt. Where exactly on mRNA the smaller subunit will get attached? The smaller subunit of ribosome will get attached to the region where the codon AUG is there, because AUG is the initiator codon, and with this particular codon only, what should begin? The process of protein synthesis or the process of the polypeptide synthesis should begin. For that sake, what will happen? The particular smaller subunit of ribosome will get attached to the region where the codon AUG is present on the mRNA. Is that clear? and AUG is recognized by the initiator tRNA. The next, what will happen? When the smaller subunit of ribosome attaches to the region of mRNA having AUG codon, to that particular region of AUG or to that particular of codon AUG, what will happen? The tRNA molecule, okay, the charged tRNA molecule will come and bind here. Okay, in the first step of initiation, what will happen? Okay, to the mRNA, the ribosomal smaller subunit will get attached. Okay, soon after the attachment, what will happen? The activated tRNA having anticodon for AUG, okay, activated tRNA having anticodon for AUG, that is UAC. This particular tRNA will come and bind to the region. Okay, it will come and bind to this region. Then what will happen? After the attachment of this particular activated tRNA to the AUG region of mRNA, the larger subunit will come and bind. After attachment of smaller subunit of the ribosome and tRNA to this particular AUG region of the mRNA, what will happen? The larger subunit will come and bind. Okay, this will forms what? The translation unit. It will form what? The translation complex. Okay, it is called as what? Translation complex will be formed. This translation complex which is formed will ultimately start doing the process of translation or start doing the process of what? The formation of polypeptide chain. That itself is called as initiation. Okay, I'll describe it once again. You know, very simple it is. In the initiation step, okay, in the first part, what will happen? The smaller subunit of ribosome will get attached to AUG region of the mRNA. Soon after attachment of smaller subunit to the AUG region of mRNA, the activated tRNA having anticodon for AUG, it will come and bind to the AUG region of the mRNA on the top. Okay. After attachment of smaller subunit as well as the tRNA to the AUG or region of mRNA, larger subunit of ribosome will also come and bind to the particular region. Ultimately, by means of formation, by means of union of all these things, okay, a particular translation unit will be formed. Right. Next step, okay, that is elongation. The second step is elongation. In the elongation, what will happen? The particular setup, that is smaller subunit of ribosome, larger subunit of ribosome, okay, they will be, okay, moving towards right side direction. That is, they will start moving from 5' end to 3' end of the mRNA. Okay, they will start moving in the 5' to 3' end of the mRNA. And as they move, okay, slowly, what will happen? The particular tRNA molecules will come here and the decoded tRNA molecule will leave its amino acid here. Okay, then the uncharged tRNA, after leaving the amino acid, it is called as uncharged tRNA. It will leave the site. Okay, it will leave the site. Is that clear? See, I'll draw the larger subunit of ribosome here. See, if this is the larger subunit of ribosome, it will have 3 regions. Okay, as given in the explanation part. See, larger subunit of the larger subunit of ribosome attached with the initiation complex, and it has 2 sites, A site and P site. Okay, actually it will be having 3 sites, that is E site, A site and P site. Okay, E site, A site and P site, EPA sites it will be having. Okay, next one, see, initiator tRNA accommodated in P site of larger subunit of the ribosome. Okay, here what will be there? In this central region, P site will be there, and here A site will be there. Okay, A site, then P site and E site. 3 sites will be there in the larger subunit of ribosome. Okay, then what happens? Initiator tRNA accommodates the P site first. Okay, first tRNA will come and bind to this region. Okay, it will be accommodated in the P site. Next, when it starts moving in the right side direction, that means when the initiator complex will start moving in the right side direction for the purpose of elongation, what will happen? Next charged tRNA will enter in the A site. Okay, and when it move further, the particular amino acid brought by the first tRNA will be given attached to the next one here like this, and what will happen? It will shift to the E site, and here by the E site the uncharged tRNA will get exit. Clear? That only is described in this particular explanation part. See here, larger subunit of ribosome, sorry, initiator tRNA accommodates the P site of large subunit. The subsequent amino acyl tRNAs enter the A site. See here, I have shown it in the diagram. Subsequent amino acylated tRNAs will enter into what? The A site, A region. Okay. Next one, the subsequent tRNA selected according to the codon of the mRNA. Okay. It is understood very clearly that according to the codons that are present on the mRNA, the subsequent tRNAs will be selected and they will come and bind to what? The particular A site of the larger subunit of ribosome. Clear? Next, the codon of mRNA and anticodon of tRNA are complementary to each other. Okay, the codon and anticodon of mRNA and tRNA respectively will be complementary to each other, and the formation of peptide bond between two amino acids, that is P site and A site, it will be catalyzed by the ribozyme. Okay, the enzyme, what? The ribozyme, it will catalyze. Okay, the formation of peptide bond will be taking place between the amino acids that are brought in P region and that is brought in the A region. Okay, see, very important point, formation of peptide bond will take place between what? Two amino acids, and where exactly the bond will be formed? Between the amino acids of P region and A region. Next, the particular setup moves to what? The next codon along with the mRNA. Okay, that will be ultimately leading to elongation of this particular chain to form a polypeptide chain. Okay, and finally when it reaches, okay, finally when it reaches a particular region where and the antisense codon is there, it will lead to the next step, that is termination. See here, elongation continues until a stop codon is arrived at P site. Okay, that means the process of elongation will be continued till what? Till the particular site or till the particular complex of larger and smaller subunit will reach a region where the stop codon arises in the P site. Okay, when the stop codon arrives at a P site, then what will happen? No tRNA can come and decode the codon because there is no tRNA. Okay, because there is no tRNA for a particular stop codon. Okay, see here, all the three steps are shown in a common diagram. In the first step, what will happen? Initiation will be taking place, and in the second step, elongation will be taking place. When after elongation, when the particular the complex reaches a region where stop codon is present, then what will happen? The process of translation will be terminated. Why? Because when a stop codon arrives at the P site of larger subunit of the ribosome, then no other or no particular what? tRNA can come and bind to that particular region. Okay? The reason is the tRNA for stop codon is not present. Okay? There is no tRNA present for any of the stop codons. Okay? There is no tRNA present for any of the stop codons. That is why what will happen? Okay? The process of synthesis of the polypeptide chain will be terminated. Okay. If there is no tRNA, then who will come and bind to that particular region? Very simple. Okay, it is mentioned here. See, when the particular P site is, okay, having antisense codon or stop codon, then instead of tRNA, because no tRNA is there for what? Decoding. Instead of tRNA, what will happen? A release factor will come and bind to that particular region. Okay, a release factor, see here, a release factor binds to the stop codon. Okay, a release factor will come and enter into the P site. Okay, when a release factor come and enter into the P site, okay, sorry, when a release factor enter into the A site, then what will happen? Termination of the process of protein synthesis will take place. See, when a release factor binds to the stop codon, further shifting of ribosome lead to separation of the polypeptide. Okay, further shifting will lead to separation of what? Polypeptide, and as a result, an mRNA also has additional sequences that are not translated. They are called as what? Untranslated regions. So before going to this particular point, I'll make clear here, I want to make you clear here that soon after the release factor enters in the A site, what will happen? All the three, that means larger subunit of the ribosome, smaller subunit of the ribosome, and the mRNA, all of three will get dissociated from each other like this. They will get dissociated, or else they will get separated from each other. Okay, by means of separating from each other, what will happen? They ultimately release the polypeptide chain, which is, okay, formed or else synthesized. Okay, ultimately they will release the polypeptide chain. Okay, so this is what the process of translation. Okay, I'd like to repeat the same thing. Okay, what is the translation? The process of formation of, okay, the process of formation of what? Polypeptide chain or protein from the mRNA template with the help of, okay, with the help of what? The ribosomes and tRNA is known as translation, or simply you can say that process of formation of polypeptide chain from the mRNA is called as translation, or protein formation from the mRNA is called as translation. Okay, translation, where it begins, that and where it takes place? The translation takes place inside the cytoplasm of the cell. Okay, during translation, what all the changes will be initiated? During translation, what will happen? In the first step, as a beginning, amino acylation of tRNA will take place. Okay, amino acylation of tRNA will take place. That means charging of the tRNA will take place, where the particular activated amino acids will get attached to amino acid binding site of the tRNA. Okay, the amino acids will get attached to amino acid binding site of the tRNA, and this process is called as what? Amino acylation of the tRNA. And very important point to note that the mRNA, okay, which is present, the sequences present on the mRNA, they will guide, or they will act as a template, or they will guide the sequence of polypeptide or sequence of amino acids that should be produced. Okay, the sequence of amino acids which are to be produced will be guided by what? It will be guided by the particular coded information present on the mRNA. Clear? And who will help for the process of synthesis of a protein? Okay, that is ribosome, which is called as protein factories of a cell. Okay, ribosomes is the protein factories of a cell. Clear? And in the ribosome, it will be having 70S and 80S type. In case of 70S type of ribosome, it will have two parts, that is larger subunit and smaller subunit. Larger subunit is 50S in nature and smaller subunit is 30S in nature. Both of these together will form 70s ribosome. Clear? And here the word or the letter S indicates Swedberg's unit. Okay, it indicates Swedberg's unit. Next, the process of translation. It has got 3 steps. Okay, the process of translation includes 3 steps: initiation, elongation, and termination. So in the first step, that is initiation, what happens? Smaller subunit of ribosome, okay, the smaller subunit of ribosome gets attached to the region of mRNA where the initiator codon AUG is present. What happens in the initiation? The smaller subunit of ribosome will get attached to the region of mRNA where initiator codon is present. Okay. Next one, after the attachment of smaller ribosomal subunit to the initiator region, okay, the initiator or else the initiator tRNA, okay, you can call it as initiator tRNA, which is charged with the particular amino acid which has got anticodon for AUG, it will come and bind to the same region. Okay. After attachment of ribosomal smaller subunit to the mRNA having AUG codon, the anticodon having, okay, or the tRNA having anticodon for this particular AUG, which will be charged with its own amino acid, will come and bind to this particular region, right? Then the larger subunit of the ribosome will get attaches, okay, to form what? The complex, okay, translation complex or initiation complex, you can call it as, right? After formation of initiation complex, the next step will be initiated, that is elongation. In the elongation, what will happen? The particular complex, which is called as initiation complex, it will start moving towards its right side. When it starts moving towards its right side, then what will happen? Gradually, the tRNA or charged tRNAs will start entering into a site of the larger subunit of ribosome. They start entering, or charged tRNAs will start entering the A site of the larger subunit of ribosome. And which particular charged tRNA will enter into the A site? Do remember, the charged tRNA of corresponding codon present on the mRNA will enter into A site of what? Larger subunit of the ribosome. Then when the complex move towards its right direction or right side direction, what will happen? The tRNA which is first present in the P site, it will go to the E site by leaving its amino acid there itself attached to the next amino acid carried at the A site. Is that clear? ಅಂದರೆ ಪಿ ಸೈಟ್ ಒಳಗಡೆ ಇದ್ದಂತಹ ಟಿ ಆರ್ ಎನ್ ಎ ಏನು ಮಾಡುತ್ತೆ, ತನ್ನ ಅಮೈನೋ ಆ್ಯಸಿಡನ್ನು ಕ್ಯಾರಿ ಮಾಡಿದಂಥ ಅಮೈನೋ ಆ್ಯಸಿಡನ್ನು ಎ ಸೈಟ್ ಒಳಗಿರುವಂಥ ಟಿ ಆರ್ ಎನ್ ಎದ ಅಮೈನೋ ಆ್ಯಸಿಡ್ ಜೊತೆ ಬೈಂಡ್ ಮಾಡುತ್ತೆ ವಿತ್ ದ ಹೆಲ್ಪ್ ಆಫ್ ವಾಟ್? ದ polypeptide, or else with the help of the peptide bond. Okay, like this. Then when it attaches its own amino acid to the next one, it will get decharged. Okay, it will get decharged, and this decharged or else discharged the tRNA will get present or will get escaped out of the larger subunit by the region called as E site. Okay, E site is nothing but what? Exit site, where exactly the uncharged or else decharged tRNAs will get out of this particular complex. Is that clear? And this process of attachment of the amino acids and entry of the tRNAs to the A site will continue, okay, up to, okay, the next step, that is termination. Okay, when the continuation has been taking place continuously, or when the continuation is being going on, then it will get continued, or it will be continued up to what? The region in the P site. Okay, in the P site, what should arrive? A stop codon should arrive. Okay. Okay, the process of elongation will be continued till a stop codon arrives in the P site. Okay, the process of elongation will continue till the stop codon arrives at the P site. When the stop codon arrives in the P site, okay, there will be no tRNA. Okay, there will be no tRNA present for what? Decoding that stop codon. Okay, instead of tRNA, what will enter? A release factor will enter in the P site. Okay, sorry, not in the P site. A release factor will enter into the A site of the larger subunit of ribosome. When the release factor enters into the A site, it ultimately help for what? Dissociation of all these three. That means larger subunit of the ribosome, smaller subunit of the ribosome, and mRNA separately. When they get separated altogether, they will release the chain of amino acids freely, which is called as free polypeptide chain. Okay, and do remember that this free polypeptide chain which is released, it will undergo several structural changes, okay, and several foldings, okay, depending upon the nature which particular amino acid sequence is formed. Depending upon that, what will happen? It will undergo several structural changes to form what? A protein molecule. Okay, to form a protein molecule, it will undergo several structural changes. And last important point to note down here is, see, an mRNA also have some additional sequences that are not translated, which are called as untranslated regions or else UTRs. Okay, an mRNA may also have some of the regions where the translation is not seen. Okay, the translation will not be seen, and that type of regions where translation is not seen, they are called as untranslated regions. Clear? And further about the regulation of gene expression and next part of the chapter we shall discuss in our next video. Right? Thank you.